मैं राजवीर सिंह शेरान एसोसिएट प्रोफेसर इन केमिस्ट्री SRR मोरका गवर्नमेंट PG कॉलेज झुंझुनू. First of all, good afternoon to all the students of MSc final students. Today we will continue the coenzyme chemistry. कल हमने 3 कोएंजाइम डिस्कस किए थे जिसमें स्ट्रक्चर ऑफ कोएंजाइम ए, दूसरा थायमिन पायरोफॉस्फेट इसकी स्ट्रक्चर, और तीसरा pyridoxal फॉस्फेट जो है इसके बारे में हमने डिस्कस किया था. टुडे वी विल कंटिन्यू द सेम टॉपिक एंड द फोर्थ कोएंजाइम इज NAD+. NAD+ means nicotinamide adenine dinucleotide. It is also known as DPN, means diphosphoridine nucleotide, or co-dehydrogenate first or coenzyme first. This is the hydrogen transfer coenzyme and its activity is due to nicotinamide. It accepts hydrogen from substrate which is oxidized and then transfer step by step to other compounds such as fluoroprotein, cytochrome B, cytochrome C, cytochrome N and cytochrome oxidase. The last compound gives its hydrogen to oxygen forming water. The overall result is oxidation of substrate. Transference of hydrogen is reversible and stereospecific as shown in the following reaction. We can see in this diagram that one important use of NAD+ is its use during the alcoholic fermentation, whereas acetaldehyde is reduced to ethanol by means of NADH. Now we can discuss structural elucidation of NAD+. So, structure of NAD+ has been revealed on the basis of hydrolysis under different conditions as given below. For example, we can take acid hydrolysis. Hydrolysis of one molecule of this coenzyme under acidic conditions gives one molecule of adenine, 2 molecules of D-ribose 5-phosphoric acid and 1 molecule of nicotinamide. So, NAD+ undergoes hydrolysis in presence of acid. It gives rise to adenine and D-ribose 5-phosphoric acid and nicotinamide. This result indicates that NAD+ contains 1 adenine, 2 ribose and 2 phosphoric acid and 1 nicotinamide molecule per molecule of NAD+. Now second, it may be alkaline hydrolysis. Under mild conditions, alkaline hydrolysis yields adenosine for diphosphate ribose and nicotinamide, but under a strong alkaline condition, adenosine diphosphate is obtained. Now, NAD+, OH- hydroxyline in strong conditions, it can give rise to adenosine diphosphate, or in mild conditions, it can give rise to nicotinamide and adenosine diphosphate ribose. So, alkaline hydrolysis indicates that adenosine and ribose molecules are linked at 5 position through pyrophosphate group. C, it may be enzymatic hydrolysis. Enzymatic hydrolysis of NAD+ produce adenosine and nicotinamide riboside, indicating that nicotinamide molecule linked to ribose molecule by C1 of ribose. So NAD+ enzymatic hydrolysis, it gives rise to adenosine and nicotinamide riboside as shown in the reaction. Result of degradative hydrolysis under acidic basic conditions and enzymatic hydrolysis conclude NAD+ in its one molecule constitute of one molecule of adenosine linked to one molecule of nicotinamide ribose through pyrophosphate group at 5 position of their ribose moieties. The structure of NAD+ is written as shown in this example. So we can take synthesis of NAD+. So synthesis of NAD+ gives by told in 1957 as shown below. We contain 1-acetyl 2,3,5 triorthobenzoyl beta D-ribofuranoside. In presence of HCl and ether, it gives rise to the compound. We can take this NAD plus as given in the reaction. Mechanism of this NAD plus act as coenzyme in oxidation reduction reactions. It in the reaction of horse liver alcoholic dehydrogenase, NAD plus is the first substrate to bind with the enzyme, which after the reaction gets converted into NADH. Dissociation of NADH from the enzyme is rate determining step of the complete reaction. For this reason, NAD plus is regarded as coenzyme and not as substrate. Alcohol dehydrogenase occurring in horse liver exists as a dimer. Each unit has 3 binding sites, one for NAD plus and 2 for zinc 2 ions. During this reaction, a ternary complex is formed, structure of which is identified by X-ray studies as given below. This coenzyme. So next coenzyme is NADP plus, means full form is nicotinamide adenine dinucleotide phosphate. It is also known as coenzyme second or phosphocoenzyme, or TPN, triphosphopiridine nucleotide, or co-dehydrogenase second. It has one additional phosphate group than DPN molecule in position 2' of the ribose molecule of adenosine. It also helps in dehydrogenation. Now you can see below the structure of NADP plus. So NADP plus on hydrolysis by a nucleotide phosphate gives nicotinamide nucleotide along with other products, which was identified as adenosine 2'5' phosphate. Selective hydrolysis of the 5' position of this group, phosphate group of the latter product, further gives adenosine 2' position. Hence the coenzyme second structure may be written as, so you can see in this the base ribose sugar here attached with phosphate, and then this adenosine unit is also joined. So next our coenzyme is FMN and FAD, means flavin nucleotides. FMN means flavin mononucleotide. It is also known as riboflavin phosphate. Riboflavin is a component of this coenzyme. Another name of this coenzyme is riboflavin 5' phosphate, and it's combined with various specific proteins of coenzymes to form enzymes, that is L-amino acid oxidase and cytochrome C reductase. The structure of this coenzyme is given below. So structural evaluation of FMN we can discuss. FMN is obtained by the phosphorylation of lactoflavin. Therefore, if we elucidate the structure of lactoflavin, also known as riboflavin, structure of FMN can be determined. Molecular formula of riboflavin is C17H20N4O6. Silver salt of riboflavin has tetraacetate upon acetylation, indicating the presence of 4 hydroxyl group alcoholic in its. Oxidation of riboflavin and lead tetraacetate gives formaldehyde, that indicates presence of 1 primary alcoholic group in it. Alkaline hydrolysis of riboflavin gives lumiflavin. Photolysis of riboflavin also gives lumiflavin. Molecular formula C13H12N4O2 according to the following equation. We can take riboflavin in presence of sunlight, it gives rise to lumiflavin, and plus some side chain product C4H8O4 is obtained. So lumiflavin can either be acetylated neither be acetylated nor it can be oxidized by lead tetraacetate. Therefore, C4 fragment is tetrahydroxybutyl side chain or CHOH, 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 CH2OH. So lumiflavin contains methylamino means tertiary amine type group in place of side chain. Therefore, the side chain must be attached to nitrogen and methylamino group. Alkaline hydrolysis of lumiflavin yields urea and amino carboxylic acid as shown in this reaction. So, it can be identified as an aromatic diamino compounds by usual text, as it gives rise to blue precipitate with ferric chloride. It must contain N-methyl orthophenyldiamine nucleus FM C7H10 and 2. So given like this type of compounds. So in this reaction you can also see side chain of riboflavin. So riboflavin upon irradiation in acid solution gives lumichrome on the basis of analytical work similar to lumiflavin, and synthesis of the following structure has been identified for it. So this alloxane structure is isomeric with isoalloxane structure, means this is the structure of lumichrome. So this completes about NAD coenzyme and NADP plus coenzyme FMN and like this. थैंक यू. आज इतना ही करेंगे. इसमें बचे हुए कोएंजाइम नेक्स्ट ही कंटेंट में करेंगे अब. थैंक यू.